DNA sequencing enables to perform an analysis of DNA because it provides the basic information of the sequence of nucleotides. Knowing this, we can locate regulatory and gene sequences, make comparisons between homologous genes through species, and identify mutations. From this data, in 1974, two methods were independently developed for DNA sequencing.
The first method is the one of Sanger-Coulson. This method is also known as the dideoxy chain termination method, based in the use of chain terminators. The dideoxynucleotides, which differ from deoxynucleotides in the lack of the 3' OH, causing the extension of the chain in the replication to stop. This allows the generation of DNA fragments of different sizes. The key of this method is that all reactions start from the same nucleotide and end with a specific base. Sanger sequencing involves making many copies of a target DNA region. Its ingredients are similar of those needed for DNA replication in an organism, or for polymerase chain reaction, which copies DNA in vitro. They include a DNA polymerase enzyme, a primer, which is a short piece of single-stranded DNA that binds to the template DNA and acts as a starter for the polymerase. The 4 DNA nucleotides, which are D-ATP, D-TTP, D-CTP, and D-GTP. The template DNA to be sequenced, and however, a Sanger sequencing reaction also contains a unique ingredient, dideoxy or chain terminating versions of all 4 nucleotides, each labeled with a different color of dye.
Steps for the process. The DNA sample to be sequenced is combined in a tube with primer, DNA polymerase, and DNA nucleotides. The 4 dye label chain terminating the deoxynucleotides are added as well, but in much smaller amounts than the ordinary nucleotides. The mixture is first heated to denature the template DNA, then cooled so the primer can bind the single-stranded template. Once the primer has bounded, the temperature is raised again, allowing DNA polymerase to synthesize new DNA starting from the primer. DNA polymerase will continue adding nucleotides to the chain until it happens to add the deoxynucleotide instead of a normal one. At that point, no further nucleotides can be added, so the strand will end with the deoxynucleotide. This process is repeated in a number of cycles. By the time the cycling is complete, it's virtually guaranteed that a deoxynucleotide will have been incorporated at every single position of the target DNA in at least one reaction. This is, the tube will contain fragments of different lengths, ending at each of the nucleotide position in the original DNA. The ends of the fragments will be labeled with dyes that indicate their final nucleotide. After the reaction is done, the fragments are run through a long, fine tube containing a gel matrix in a process called capillary gel electrophoresis. Short fragments move quickly through the pores of the gel, while long fragments move more slowly. As each fragment crosses the finish line at the end of the tube, it's illuminated by a laser, allowing the attached dye to be detected. The smallest fragments crosses the
Alan Maxam and Walter Gilbert developed a method for sequencing single-stranded DNA using a two-step catalytic process involving piperidine and 2 chemicals that selectively attack nucleotides: dimethyl sulfate for purines and hydrazine for pyrimidines. The general steps for these methods are: first of all, you have to have one strand or double strand DNA sample. Then you have to separate the homogeneous single strand DNA and labeling with a radioactive of the 5' with 32 phosphate. The second step is the modification of the base. Here's where the dimethyl sulfate acts because it takes the sugar from the base. This is where the methyl attached to the bases that are marked, and here is where the elimination of the modified base is going to occur. After the modification of the base, is where the piperidine is going to act, and it's going to fragment the DNA chain. The aim of this method is to have several differently sized DNA strands so we can analyze them in a fragmented sequence by electrophoresis in a polyacrylamide gel.
In electrophoresis, fragments obtained from the four reactions are electrophoresed. In autoradiography, the gel is exposed to X-ray and series of bands are obtained. This method is used for footprinting and epigenetics for DNA manipulation. It required working with neurotoxin hydrazine and radioactive materials. Many steps required a lot of precision, and short sequence was obtained in many days. Besides the development of other techniques, chemical sequencing lost its popularity. The automation of this method uses solid phase chromatography for DNA purification and a different way for cleavage. Rate of sequence acquisition is increased, data is more reliable, and it can perform reactions in 8 fragments at a time. It is more accessible, especially in large-scale sequencing projects.
The reason why Maxam and Gilbert method lost popularity were because of time consuming. Using this method, you could only confirm about 200 to 300 bases of DNA every few days. Also require working with large amount of radioactive material and working closely with hydrazine, which is a known neurotoxin. The development of other techniques and the simplification of the sequencing caused chemical sequencing to lose its appeal.
Sanger method has become outdated. Automated sequencing has become developed so that more DNA can be sequenced in a shorter period of time. With the automated procedures, the reactions are performed in a single tube containing all 4 DDNTP, each labeled with a different color dye. In automated sequencing, the oligonucleotide primers can be N-labeled with different color dyes, for each DDNTP. These dyes fluoresces at different wavelengths, which are read via a machine. As in Sanger's method, the DNA is separated on a gel, but they are all run on the same lane as opposed to 4 different ones. Since the 4 dyes fluoresces at different wavelengths, a laser then reads the gel to determine the identity for each band according to the wavelength at which it fluoresces. The results are then depicted in the form of a chromatogram, which is a diagram of color peaks that correspond to the nucleotide in that location in the sequence.